The Tiles and Positions module in ZenBlue allows you to create and acquire tile regions with high precision. If the microscope imaging system is not calibrated properly, you might get less than desirable results. This video will show you how to properly check the orientation and alignment of a digital camera for use in tiling experiments. This video assumes that you have the camera and microscope hardware connected to the user PC, and Zen is installed with a proper MTB configuration. Verify that the microscope components and camera are functioning properly in Zen. There are three main verification steps in this calibration procedure. The first step is to verify that the orientation of the image through the eyepieces matches that of the camera. Second, we'll verify the stage motion in each axis and invert them if necessary. Lastly, we'll ensure that the camera rotation is properly aligned with the stage movement. Place a sample on the stage and adjust the light path settings to get an image through the eyepieces. It's best to find an area of the specimen where the orientation of the field of view is easily recognizable. Send the light path to the camera and open a live view in the Zen Locate tab. Snap an image and make note of its orientation. Switch the light path back to the binocular tube and compare the image you see through the eyepieces with the image on the screen. If the images match, this step is complete. If the images don't match, navigate to the camera window and expand the model-specific section. Under Orientation, choose a different option from the pull-down list until the live view orientation matches the image through the eyepieces. Common orientations are Rotate 180 for side port cameras on inverted microscopes, and Original or Flip horizontally for binocular tube cameras on upright microscopes. For AxioCam cameras, this is assuming the camera label is facing towards the front of the microscope. When acquiring tile regions, it's very important that the software can move the stage in the correct direction of travel. If this is not the case, you may find that the tiles do not appear to be taken in the correct order. Acquire a tile region and examine the resulting image. A 3x3 tile region is sufficient. If the tiles are arranged in the proper order, you may skip to the next step. Otherwise, there can be three possible outcomes. Rows of tiles appear aligned but are acquired in reverse, columns are acquired in reverse, or all tiles are scrambled. Make note of the outcome. Now exit Zen and start MTB config. In the motorized stage component, check or uncheck the boxes under axis directions based on the outcome of the previous step. If the box is already checked, then uncheck it, and if it's unchecked, check it. For situations where the rows are reversed, uncheck or check the Invert Y-axis box. For situations where the columns are reversed, uncheck or check the Invert X-axis box. For situations where the entire image is scrambled, uncheck or check both the Invert Y-axis and Invert X-axis boxes. Remember, if the box is already checked, then uncheck it, and if it's unchecked, check it. Now press the Apply button on the bottom of the window, then press OK. Restart Zen and acquire another tile region to verify that the stage axes move in the correct direction. Acquire a tile region and examine the image carefully. You may find that features which cross the boundaries of tiles may appear to be sheared. This phenomenon occurs when the physical rotation of the camera is not properly aligned with the motion of the stage. When images like this are stitched, you'll find a stair-stepping effect along the borders of the tile region. If the camera rotation artifact is severe, proceed with this step. Open a live camera view and find a feature within the field of view that can be easily discriminated from the rest of the image. Small punctate features or the edge of a tissue section or cell body are useful for this purpose. move this feature to the top left or right edge of the image. Using the stage controller, move the stage only along the x-axis. Do this so that the feature moves towards the other side of the image. The feature should hug the top edge of the image as it moves along the x-axis. As it is moving, it should not appear to move up or down relative to the field of view. The feature will slant up or down when the camera rotation is not aligned with the stage. If the feature appears to slant up or down, loosen the C-mount adapter fastening ring or set screws and rotate the camera against the apparent slant. Tighten the ring or set screws and check the movement of the feature along the x-axis once again. Make repeated adjustments if necessary. Be aware that tightening the C-mount fastening ring may introduce further rotation of the camera. Make sure you pre-compensate for this slight rotation before you tighten it. Acquire one last tile region to verify that all the adjustments are correct. If the orientation, order, and rotation of the tile seem to be correct, but the tiles still do not seem to align properly, then you may be having an issue with scaling factors. If the theoretical scalings are not calculated correctly, the stage will appear to move too far or not far enough whenever it moves to an adjacent tile. Please ensure that all objectives and optivars are properly assigned for their respective positions on the TFT. Also, make sure that the appropriate C-mount adapter for the camera is specified in the MTB. In rare instances, you may have to adjust the stage's step size in case the stage is run by a third-party stepper motor controller. After making any adjustments, restart Zen to ensure the tile regions are acquired properly.